Though in the previous modules, system. So I just want to throw more light on this molecular diagnosis. Like the number one point we have here is that molecular diagnosis tend to it actually target the nucleic acid instead of the protein. So because protein are the byproduct of these genetic materials. And it actually consists of binding of nucleic acids to its complementary target in the nucleic acid sequence. So, and it's actually designed to actually detect changes or any aberration that occur at the DNA level or the RNA level of any human genetic material or any organism, because you can still do it you can still do molecular diagnosis for other living organisms. Then DNA and RNA extraction is actually the basis of this molecular diagnosis, because you cannot analyze DNA or RNA with multiplex PCR machine or real-time PCR machine without having done DNA or RNA extraction. Then this DNA extraction that we are talking about is equally known as DNA purification. So it's purification of DNA. And there are two methods of DNA and RNA extraction. You can either use manual method or the automated method. And both of them, they undergo the same processes. So there are different methods of this DNA, DNA molecular diagnosis. Number one is enzymatic cleavage of the nucleic acids. Then we still have a gel electrophoresis. We have application of target sequence, which is the PCR process. Then we have hybridization process with the nucleic acid probe. So this one, you can find it when you are doing particularly phenotyping. You are doing phenotyping. So these are methods of molecular diagnosis. Then for us to understand DNA extraction and RNA extraction very well, it's important that we actually understand the nature of this DNA and RNA. It's just a kind of a review of our theoretical knowledge on genetics. So we need to know that this DNA that we are talking about, they store human genetic information and they dictate the amino acid sequence of the peptide and proteins. That is why we said that this peptide or protein, they are actually the byproduct of this DNA, and they are made up of two strands of nucleic acid first. Then they have a helical structure, which is stable as a result of many hydrogen bonds between the pairs, and they are hydrophobic, and they have a hydrophobic interaction between them. This bond can be broken and strand denatured and separated. So that is the principle behind this last point. Is actually the principle behind the PCR process. Then the nature of the RNA. So just like the DNA, here RNA is just similar to DNA, but the difference is that the RNA have a single strand. It's a single strand, and the ribose as sugar in the RNA. Then we have a uracil that replaced thymine as a result, as a purine basis, because we are what make up this DNA and RNA. We have a pyrimidine and purine basis. Then we equally need to understand that working with DNA to synthesize protein, denatured DNA binds to RNA and reanneal to reestablish original DNA, which means when DNA separates, what you will now have only one strand, which can be likened to RNA, but at the end of the day, another complementary sequence actually anneal to form the DNA again. So that is why they are similar. Then what are the materials that we require for this extraction? Be it manual, for instance, manual process. So in the manual process, we actually need micro centrifuge machine. We need the spin column. If you look at this pictograph, this one is the actual the micro centrifuge. Then this one that I'm pointing out, this green, something like greenish, is what we call spin column. While this one with blue background is what we call collection tube. Is what we call collection tube. So, and we need biosafety cabinets. We need prepared and prepared tip. We need a vortex mixer to achieve this process. Then we need the sample. We need the sample. The sample could either be blood sample, whole blood sample, or plasma, or you have a buffing coat. So the ideal is either plasma or buffing coat. So this plasma or buffing coat, they are rich in DNA. So when you are contemplating using blood, so you either go for either using buffing coat for DNA extraction or using plasma. So they have higher yield than the whole blood. Then you need the reagents, which is the lysine buffer, the wash buffer, the elution buffer, or the still water. You equally need a water bath. So these are the materials that is required for this DNA extraction. Remember, in one thing I need to emphasize here, in molecular diagnosis, I believe that some of my colleagues have actually threw a little light in this aspect I want to talk about. So for you to achieve a molecular diagnosis, you need a clean chain laboratory system. A clean chain laboratory system is talking about where you prepare your DNA and where you extract your DNA should be different from where you actually amplify and analyze your DNA samples. So this is what we call a clean chain laboratory system. then what are the reagents that is required? For instance, in this particular lecture, where we are going to be relying on Crygene products, because there are other products that produce reagent that you can actually use for DNA extraction manually. So Crygene, they have reagents that you can use manually. They equally have the one that you can use for automation. So in preparing your reagents, how do you achieve this? Like reagents, most of them come in like lyophilized form, which means you have to, and they have a diluent, which you reconstitute to have your working reagent. Then first, you need to add 12 ml of this protease solvent into the lyophilized Crygene protease or proteinase K. Then you add absolute ethanol to buffer AL. You will see this reagents that I'm talking about. If you are using this Crygene product, all of them come in a pack. You mix thoroughly by shaking. Then there's buffer AW1, which is supplied as a concentrate. So before using for the first time, then what you need to do is you need to add absolute ethanol to the buffer. Then the same thing with buffer AW2, you need to equally add absolute ethanol to concentrate it. Then after preparing all these reagents, they are very stable for 1 year at room temperature. So you can use it for 1 year. That is if you don't have a high volume of work. Then all the process of DNA follow this 4
Hello. Can you unmute your mic?
Sorry.
Yes, I think you need to start on the previous slide. So your mic was muted. Yes.
Okay.
I think
So like I was saying, so I said that the purification process or extraction process follow this first stage. Like I said, the first stage is actually what happens is actually the destruction of the cellular structure of your sample, which means it's more like a disintegration or lysing of your sample to release. Second stage, which is washing and dry and binding, is actually the separation of your DNA from the contaminants, because as the reagents disrupt the cellular structure to release the DNA. So the other part of the sample, they contain protein and other, which is not needed. So that is where this second stage is, that is washing and binding. So then the binding that we are talking about is, by the time you pour this sample into your speculum, there's what we call a silica membrane that is part of the speculum. Or if you are using automated process, there's what we call magnetic beads that is part of the automated machine that actually binds a kind of bind this DNA or RNA. So then this test stage is the washing and drying. So at this stage, maybe you are using your wash buffer AW2, and at this stage, it removes the remaining contaminants and at the same time dry the sample, the DNA template. Then the last stage is where you release the DNA from the speculum or the magnetic bead using elution buffer or DNA-free distilled water. So I was explaining, I said manual process using cryogen mini kits. So what you need to do, there is what we call cryovalve. There are different sizes of cryovalve. We have 2 min cryovalve, we have 5 min cryovalve. Using 2 min cryovalve, for instance, you prepare your reagents, which is your Proteins K, into this 2 min cryovalve. Then you now add your sample. If you are using blood sample, you add 200 microliter of your either buffing coat or plasma, because I said that it is advisable to use plasma or buffing coat for you to have high yield of DNA or RNA, as the case may be. Then after which you add of this buffer buffer AL, which is part of the reagent that we've already discussed about. Then after which you mix thoroughly, then you incubate this sample for 10 minutes at 56 degrees centigrade using that water bath. That's why we listed some of the things that you needed for this extraction process. Then after which you add 200 microliter of absolute ethanol to this sample or this mixture already in your cryovalve. Then you mix and vortex. Then after which you prepare the mixture. Either you prepare the mixture into the speculum that is already placed in a collection tube, or you pour it into the spin column that is already placed in a collection tube. I think I show us some pictograph of this collection tube and spin column. So what happened is once you pour it, the silica membrane in this spin column have affinity for DNA. So it actually binds the DNA, which means it traps the DNA. Then while other that is in that spin column. So then you centrifuge for 1 minute. Then after centrifuging, you remove. Then you discard all the content that is collected in the collection tube, which is regarded as the contaminant. Then after which you place this spin column into a new collection tube. Then that is when you now add your buffer AW1. Then you centrifuge at 14,000 RPM for 1 minute. Then after which contaminant that is collected in the collection tube, then you place this spin column into a new one, a new collection tube. So like this, at this stage, the first one is washing and binding. Why this step I'm trying to explain, that you place this spin column into a new collection tube and add a buffer AW2 is the washing and drying stage. Then after adding your buffer AW2, you centrifuge at the same 14,000 RPM for 3 minutes. Then you discard the contaminant that is collected in the collection tube. Then to ensure that it's dried, you can still centrifuge for a minute. After placing that spin column into a new collection tube to ensure that it's thoroughly dried. Then after which you place this spin column into a new collection tube. Then you add 200 microliter of your buffer AE, which is your elution buffer. Or if you don't have this elution buffer, then you can use a DNA-free distilled water and incubate for 1 minute at room temperature. Then you centrifuge for a minute to elute the DNA. Then after which you now discard the spin column, and then the collection tube is now having your elute, which is regarded as your DNA template. So you don't need to discard that one. Once you get to elution, the content in that collection tube becomes your DNA template. So you save. If you want to use it immediately, you use. But if you don't want to use, maybe you want to use it in 2 days, 3 days, a week time, you can store at -20 degrees centigrade. But for longer time, for years and all that, you can store at -80, -degree. So the same procedure is actually used for extracting DNA from either tissue, bony tissue, any other thing, as the case may be. You can extract DNA from from hair, from any part of the body. So what you need to do, for instance, if you are using tissue, for instance, if it is a bony tissue, the first thing is you need to decalcify. But if not a bony tissue, what you need to do is to kind of pound this tissue, at least to break it into bits. Then after which you add your PBS. PBS is phosphate buffer saline. So like during the time of COVID, this phosphate buffer cell line was replaced with VTM. So it's still the same thing, they do the same function. The difference between this PBS and that VTM was that they added some antibiotics. So after pounding, you add 80 microliter of your phosphate buffer into like 100 milligram, 10 milligram of pounded tissue. Then after which you add 100 microliter of buffer ATL. This buffer ATL is specific for tissue when you are extracting DNA for tissue. So you will still see it in this kit in case you want to extract from tissue, in case you want to extract for any other thing. So it's there, so you will still see them. So it's just for you to pick the one that you want to use. Then you now add 20 microliter of proteinase K. This proteinase K, that is where that of blood starts from. So you add 20 microliter of proteinase K, and the function of this proteinase K is that it has what? It has a lysine property, or something that a property that can disintegrate. It means it is able to break down protein and release the DNA. So that is why it is called proteinase K. So then you voltage and incubate at 56 degrees centigrade for 1 to 3 hours. In between this time is enough for you to achieve this disintegration stage or lysine stage to actually release the DNA into the whole content, which is both buffer ATL and PBS that is there. Then after which you now add 200 microliter of buffer AL. Remember this one is still the same process as in the case of the blood. Then you mix for 15 seconds and incubate at 70 degrees for 10 minutes. Then after which, then you now add 200 microliter of absolute ethanol to this sample. It could be 96 or 95. Let me say 96 to 100, 100, which is the absolute ethanol. Then after which you mix for 15 seconds. Then after mixing for 15 seconds, then you carefully decant all this mixture into the spin column. That is the liquid content of it. You carefully pour it into the spin column that is already placed in a collection tube for a minute. You centrifuge for a minute, then you discard the contaminants, just like we are explaining in the case of the blood. Then after which you add the same volume, that is 500 microliter of buffer AW1 for washing and binding. So after everything, after adding, you centrifuge at 14,000 RPM for a minute. Then you discard the contaminant and the collection tube. Then you now place the spin column into a new collection tube. You therefore add the buffer AW2 and centrifuge for 3 minutes at the same RPM. Then after which you remove, you discard the contaminant and the collection tube. Then you place this spin column into a new collection tube. Then it is now time for elution. You now add 200 microliter of that elution buffer, buffer AE, or DNA-free distilled water. The reason why we say DNA is free, because we don't want anything to interfere with the DNA of the sample that you are looking at. If you use a distilled water that contains DNA, which means during your amplification process or your PCR process, which means there may be false positive or false negative result at the end of the day. So, and depending on the concentration of the DNA that you intend to get, so that is equally a function of the amount or the volume of elution buffer that you are going to add. If you need a more concentrated DNA, which means you are going to use less of the elution buffer, which means you may either use 100 or you use 50. So as the case may be. So then after elution, then you either store your elute or you analyze your DNA templates immediately. So this is how to extract DNA from tissue and also blood. Then the same thing is the same way you can equally extract DNA from either hair or any other material that you want to extract DNA from. So like in the hair, if you are extracting DNA from the hair, remember from that hair, you are going to put PBS for some time. Then after which you now add all this your reagent to actually release the DNA. Then you add all your wash buffer for washing and binding. You add your elution buffer to elute. It's this same method, it's same process. Then for RNA extraction, for instance, during the pandemic, COVID-19 is an RNA virus. So, and the procedure is just like, okay, you are extracting DNA from swab or bar body. Bar body, for instance, you scrape use swab stick to scrape the chip, inner cheek of the mouth. So that is if you want to do paternity testing. Most times that is used for paternity testing. So you use bar body to do paternity testing by using that swab stick to scrape the cheek. Then that one is still the same process like COVID-19. That you swab the nasopharyngeal and the oropharyngeal, then you now put it in VTM. That's for COVID-19. So in this case, if you are putting it like most cases for paternity test, they may not put it in VTM or PBS. They may just bring a swab stick that contains a swab of anything. So all you need to do is to put it in PBS to efflorate the cell into the liquid. Then therefore you now initiate your purification process for either DNA or RNA, as the case may be. But for COVID-19, we are focusing on RNA because it's an RNA virus. So this swab stick that is actually taken is placed inside the VTM. So all you need to do is to add 80 microliter of that sample now. that is the VTM, the content. Either if it is PBS, you take 83 microliter of PBS and put it into your 2-min cryovalve or 5-min cryovalve. Then you now add, for instance, like this kit is RNA mini prep kit. Most of the kit that is actually used for this manual RNA extraction for COVID was like RNA mini prep kit and the likes of them. There are many brands. So then you now add 240 microliter of trizol and voltage to mix, and you will incubate for 5 minutes at room temperature. Then after which you still add 240 microliter of your absolute ethanol to the lyse sample that is already in that cryovalve. Then after which you now transfer the whole content into a spin column that is placed in a collection tube. Then you place the tube into a microcentrifuge and spin at 10 to 16 grams for 30 seconds. So then you remove and discard the contaminant, which is the content in the collection tube, and place the spin column into a new collection tube. Then you now add 400 microliter of this direct, they are part of the kit, direct Zol RNA pre-wash, and you centrifuge for 30 seconds using this micro centrifuge. Then after which you remove and discard the contaminant which is inside the collection tube, both the collection tube and that content. Then you now place the spin column into a new collection tube. Then you now add another 400 and centrifuge for 30 seconds. So you are repeating it twice. And then after which you discard the contaminant and you place in a new spin column. Then you now add 700 microliter of RNA-free wash buffer. Or then once you place it in that, once you add the RNA-free wash buffer, which is part of the reagent, you centrifuge for 2 minutes, then you transfer it into a new spin column. Then you now add your elution buffer. You can add 200 microliter, you can add 50 microliter, as 100 microliter, as the case may be, depending on the concentration. So after which you can now put it, maybe you put it in a since you have collected it in a collection tube, you can, if you want to store, you can transfer it into another cryovalve, or maybe if you have a rack that can preserve that collection tube, you will store, depending on the time interval that you want to analyze. So this is how to extract RNA. Then DNA. So the automated method is just machine. We have different machines for extracting this DNA or RNA. Like we have what we call Magna Pure, we have cryosiphinis, so many of them, the likes of them that can do this extraction. So then what you need to do is just to actually, just like the conventional automated machine that we have, for instance, chemistry machine, most of, not semi, this one is, because we have a semi-automated machine for chemistry and we have full automated machine. Like some of these automated machine for extraction of this, they are they are full automated machine. So it's just for you to roll the reagents into the reagent compartment, put the prepared in the prepared compartment, then the sample in the sample compartment, then you that is after reporting into the maybe a tube that you want to place in the sample compartment, then you place them, then you put everything together, then you select the parameter on the screen that, okay, I just want to do extraction for DNA or RNA. Then you press run. And once you press run and the process is complete, it's just for you to remove and store or use to amplify or analyze. So this is the method for DNA extraction. Then there's an exception of this manual method and automated method. There are some machine that doesn't require you to do this either automated DNA extraction or manual DNA extraction before you analyze you do your molecular analysis. For instance, GeneXpert is one of them as an example of it. Then we currently have Abbott machine that is used for viral loads. Then we have Roche machines that equally does this. Then I think some Cobas can actually do same. So they will analyze it. They will do not that the extraction is excluded. They are imputed together. They perform extraction. They perform the analysis. They extract, they amplify, and they analyze. So that is everything is inbuilt in that in those machines that I just made mention of. So then, unlike mobile too, mobile, there is one that extracts, there is one that actually analyze. So that one is a fall within this category too. But I just want us to know that there are some machine that you don't need to do any extraction. Just place your everything into this your sample into these machines. They will do the extraction, they will do the amplification, they will do the analysis for you. Then all you need to do is just for you to program the machine to do all this. Program the machine, and the machine will give you what you want. So that is it. Then some of this machine, how do you quantify your DNA sample or RNA sample? There are some machines that can help you quantify this the concentration of this the DNA or RNA template. We have what we call nanodrop. Nanodrop is used to quantify it, but some PCR machine can still do the same work because there's some inbuilt software inside that does this concentration calculation of the concentration in the machine. So this is actually the end of DNA extraction. So but I will not end this particular session without discussing PCR process, because after you amplify, there's need for us to discuss some little steps we need to take in order to achieve your results. So the PCR process, what happened is that there are things we need to have to actually achieve the PCR process. It is actually called the master mix. So it means you must have to prepare the master mix for you to actually achieve it. What we call master mix, they are the precursors that actually help to amplify. What PCR actually does, it is called polymerase chain reaction, which means it actually does the photocopy. Hello? Hello? Are we together? Are we together?
Yes, sir, we're together.
Yes, sir, we're together, sir.
Okay, what the PCR actually does is actually to photocopy your DNA. That is a target nucleic acid. For instance, a target DNA. For instance, I just want to use a chromosome to analyze this. We have, we have different type of chromosome. We have chromosome 5, we have chromosome 1, chromosome 2, chromosome 3. So for instance, in DNA analysis, you are targeting one specific DNA. So maybe in that DNA, you are targeting a genetic information that is present at chromosome 5. So you are amplifying that chromosome 5 to get millions of copies of that chromosome 5 to make it detectable. It's just to make it detectable, because there are some quantity that is detectable by the machine. If it is less than that particular quantity, which means the machine may not be able to detect it. For instance, if you have done, for instance, let me say TB culture, for instance, if you have done TB culture, let me use TB culture as an example. For instance, maybe the first time you did gene expert for that TB sample, for instance, either sputum sample, then maybe you may have, they will tell you, okay, MTB detected trace. By the time you now culture that sample, it means that after culturing the sample, what that culture helps to do is to amplify the number of the bacterial load in that particular sample. Then after which, if you now take that same concentrated, that same the growth of that sample and do DNA and do gene expert on it, you will discover that it may either tell you medium, MTB detected medium, or high, which means it has amplified to an extent that it can now give you the accurate result that, okay, this TB is viable or is not viable, because there are some, at the end of the day, there will not be growth. It means they are not viable. They are just dead TB. For instance, I'm just using it as a case study in this illustration for this amplification. So the same thing is what is happening here. So you are actually making many copies that can be detectable by the machine. Excuse me. So this master means they are just the precursors that make it happen. So number one, we have the reverse transcriptive enzyme. They are just enzyme because it's enzymatic method. So we have the DNA polymerase enzyme. We have the nucleotide, which is the forward and the reverse primer. We have the probe. We have this magnesium chloride. They are acting as a kind of a catalyst, or more like a catalyst in the process. Then we have buffer. So what you need to do is to reconstitute, like in this master mix, it comes in a pack. It's a reagent too. It comes in a pack, but you have to reconstitute them, prepare them, because in this thing, there is a kind of procedure or protocol that comes with the reagent that will tell you, okay, add so volume of this reverse transcriptive enzyme to so that you get the perfect volume for your PCR process. So after preparing this reagent, you now have what we call master mix. So this master mix is what you use to amplify your DNA template or DNA extract. So what you need to do, the volume of the master mix must be higher than the volume of the DNA or RNA extract. So which means when you are taking 16 microliter of master mix, you are adding just 40 microliter of the DNA or RNA extract. So after which you mix and load this mixture into the PCR microplate. There's all PCR has a microplate, just like you are doing enzyme, you are doing a hormonal assay. So you know there are so micro plates wells. So the same thing for this PCR. So you place, after putting this mixture into the PCR micro plate, then you place the plate into the PCR machine. It may be multiplex, it may be real time. So then after which, then you go to the run, you program the machine. So, but when you are doing this PCR amplification, this step, this bullet that I said you go to run, like in the practical session, you will just see go to run, but you will not see this programming. But I'm just putting this programming for you to know what is inside, what is happening in the this process that the machine is undergoing was programmed. The program in that machine, for instance, if you are doing RNA analysis, this is the program you will see. You first of all see 50 degrees centigrade, 50 to 56 degrees centigrade. So the reason for that is where annihilation occur. Annihilation in the sense that because RNA is a single strand, so there's a need to do a revised transcriptase. So for a revised transcriptase enzyme to be added so that they can transcribe it to DNA. So that is the primer will now be added at that either at 5' or at 3' to make it to amplify it to DNA before denaturing it to release the strand to form a single strand, so that another annihilation may occur that will attach to the first strand. Another one will still annihilate to the second strand, so that it will give multiple copies. You know, 1 will give rise to 2, 2 will give rise to 4, 4 will give rise to 8. It's just like that. It's a multiple process. Then after the annihilation, then the annihilation process is programmed most time for 15 minutes. Then the denaturation process is this 15 the 95 degrees centigrade. Denaturation, because since you are working with RNA, after it has formed a DNA, so there is need to break that DNA down to separate the strand, so that, because one have produced a single strand of DNA. So after which, that strand will not be broken to release the strand to make it 2 RNA strand, so that annihilation will occur that will now give rise to 2 DNA templates. Then after which, that 2 DNA will not be disintegration again at this 95 degrees centigrade. So denaturation will happen to that 2 strand, which will now separate it into 4, for instance. As the 4 separates, so meaning there will not be annihilation of the 4. That 4, it will not be 4 strands. Then at the end of the day, denaturation will occur again to separate that 4 into 8. That is how the process goes. And the machine will now be programmed for a number of cycles, either 30 cycle, 40 cycle, 50 cycle, 45 cycle, but the ideal cycle is between 40 to 45 to be able to make a billions of copy of DNA or RNA. Then the last program is 60 degrees centigrade for 30 seconds. So that is the extension process. So because there's annihilation, there's extension, there is a denaturation, there is extension for RNA extracts. So that is when you are amplifying RNA extract or templates. But when you are programming for DNA amplification, the program is different. The first program that you see for DNA is that the machine will be programmed first for either 95 to 96 degrees centigrade. First. Why? Because it's a DNA template that need to be denatured to release the strand, to divide the strand into two. So that is why for that to occur, it occur between a certain temperature, between 95 to 96 and 98, as the case may be. Then after the separation, which means the machine will be programmed to allow annihilation to occur. And the second program for DNA is between 50 to 56 degrees centigrade. That is when that is where annihilation occur. So after which you program for the number of cycles that you want the machine to undergo. Then the last one is you program the time it will take for extension to occur, because the extension process is very short. It's very short. That is why you program for like 60 degrees to like 62 for extension to occur. And it takes like 30 seconds. So after which you run the cycle and you save the results. So I just, after you go to the run and you press run, what will happen is that the machine will start amplifying, after which you save the result. At the end of the run, you click analyze and you get your results. So we have different thresholds at which this thing is taking place. So we'll still talk about them as we progress. So the PCR process that I just explained is these 4 stages. In some cases, they will tell you it's 3 stages. But for me, because of what happened, the initialization process is the stage at which all the contaminants are actually released. Then there is activation of the DNA polymerase and denatured other contaminants to actually occur. So this thing happened between a time interval of 2 to 10 minutes. So, but most literature will not tell you of initialization, or most people will not tell you about initialization. They will tell you denaturation, annihilation, and elongation or extension. So at the denaturing stage, like for DNA, is where there is a breaking of the hydrogen bond that held the 2 helical structures together to release the strands. So, and it happened between 94 degrees to 98 degrees centigrade. That is why I said the first program for DNA is between 94 to 98, as the case may be. So, and after which annihilation is programmed for between 50 to 56 degrees to enable primer to bind to the complementary target sequence. For instance, either the RNA template and all, as the case may be. So then after which extension starts. After the primers bind to either 3' or 5', then extension start or elongation start. So, and it take a shorter time for you to achieve this. So this is the PCR process. So there are important factors that actually affect this PCR process, and which we need to take note in order to ensure we avoid false positive or false negative results. So these are some of the factors that may interfere with your PCR process. The composition of your DNA template. The second one is the DNA polymerase choice that you take. Another one is your buffer component. Another one is your primer design. Another one is the additive or inhibitors. For instance, I said during the preparation, I said for you to achieve PCR, you need to prepare master mix. I made mention of magnesium chloride, which is a kind of additive that tend to inhibit some process too. So when we are talking about composition of the DNA template, we are talking about what does this template made up of? Does it have high is it a CG template or AT template? When we are talking about that, we are looking at the purine and the pyrimidine basis. The purine and the pyrimidine basis. When you are looking at is it AT-rich template, because adenosine tend to bind with thymine, while cytosine tend to bind with guanidine. So if it is cytosine guanidine, it means it's a long chain. It's a long chain. But if it is adenosine and thymine, it means it's a shorter template. It means it's a shorter template. So when the precursors are adding, which means it takes shorter time for annihilation to complete. But for the CG template, which is a cytosine guanidine, it takes a longer time because of the long nature of the template. So there may be a problem. This may result to some certain kind of problem. So it means sometimes there may be incomplete separation for G-series template, and in terms of the A-series template, there may be a shorter time of lack of specific binding of this primers to either the 3' or 5'. So I don't want to digress further into this composition, but just understand it that most times, because of this, either the shorter or the longer, there may be incomplete separation, there may be a lack of a specific binding to the primer. Then we are not talking about the second one is the DNA polymerase choice. So there are some polymerase that are not stable, while some are stable. Like type polymerase, DNA polymerase, they are very stable to heat because you know the principle of PCR machine is thermal regulation, which means it is actually increase temperature, decrease temperature for all this process to occur. So that is actually the principle at which PCR machines are operating. So then buffer component can actually equally affect the reaction. Then another one that I would like to throw a little light on is the primer design. So the primer design, we have the ideal length of any primer should be between 18 to 22. Between 18 and 22, but if it is longer than that or shorter than that, there is a problem. So which means it may lead to primer dimer. When we say primer dimer, it means that, okay, instead of this primer to anneal to either the DNA template or RNA template, they anneal to their self, which means at the end of the day, they amplify their self. At the end of the day, you now have, instead of them amplifying the DNA or RNA template, they amplify themselves, and it may lead to false positive or false negative results. Then the additive, for instance, that is why we will say you always troubleshoot all your procedures, starting from collection to, because I know that we must have discussed collection of some of this sample. Starting from collection, you need to quality control. If you do wrong collection, it may affect this PCR process. Then if you do long extraction, it may still equally affect. Then the additive that is part of the reagent you use. For instance, you collecting a blood sample. Let's say, I just want to use EDTA as an example. You are collecting a blood sample that you want to use for DNA extraction. For instance, a bottle that is meant for 10 ml sample, you just use that to collect 5 ml instead of 10 ml, which means there are still excess anticoagulants in that sample, in that specimen bottle, that could actually inhibit the reaction of this PCR process. So that is why what we are talking about by some of these factors that can actually affect this PCR process. So what you need to do to avoid this issue, more like quality control, you need to always troubleshoot all your procedures, all your steps. For instance, during purification, during DNA extraction, because purification is the same as DNA extraction. When you notice a color residue in your spin column after elution, you know that there's a problem, which means you need to repeat the process, start the extraction afresh. And to ensure that primary damage does not actually contribute to signal or poor efficiency, you need to what? You need to quality control or check the length of your primer, check whether it's within range, within 18 to 22. If it's beyond that, you know that you need not to use that primer. Then you take steps necessary to maintain primer and probe stability. So, like the one I failed to mention, when I was talking about the composition of the DNA template. For instance, like the CGRE template, they can actually there's what we call self primer. There's primer dimer, there's self primer. This because of the length of of the CGRE template, there is possibility of self-primer. This primer will not, the thing will not complete, it will not complement their self. So which can actually affect the PCR process. Then another thing is make standard core validation to find the final step in the process. The standard course is just more like a, I said, for you to quantify the concentration of your DNA, you need what we call nano drop. But this nano drop is mostly used in research facility, not in the PCR lab or normal molecular diagnostic lab. Most times you find these things, this nano drop machine in research laboratory. Then, but I said PCR can do the same process. So there's a feature which is the standard cup that is actually imputed in that PCR machine, which can actually do validation and do what we call concentration of the DNA or RNA sample. Then you should understand that that the efficiency below 90 will be addressed, different from efficiency above, for instance, 110. So you verify this setting. For instance, that is programming of your instrument. So most times you need to verify that, okay, since I'm analyzing DNA, so is this machine program for DNA analysis amplification? Is this machine program for RNA? So you need to actually verify and adjust them. So this is a way you can troubleshoot. So then application of this PCR. So we understand that it can be applied in forensic, can be medical, for instance, detecting any aberration, any presence of virus and all that, in agriculture and even in genealogy, then even in paternity testing, then even in crime detection, which is forensic. Those are where you apply these molecular diagnostic or PCR processes. So at this point, we have come to the end of this class. So if you have any question, you can ask. Thank you for your time. Hello? Hello?
Thank you, sir.
You're welcome.
Thank you very much, sir.
You're welcome. So question. Any question? Nkechi? Any question? Japhet? Any question?
Sir.
Okay.